Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. I know you're waiting for this video lecture from very long. So we are going to start the histone modification series. So we will learn about different histone modifications like histone acetylation, histone methylation, histone phosphorylation, all of them in detail one by one. So before learning about that, we will start about the brief overview, what we are going to learn throughout the consequent 5 to 6 lectures, five to six video lectures. In I guess, yeah, we will need 5 to 6 video lectures to complete whole histone modifications. So before learning about histone modification, let's try to understand about chromatin modifications. So you have learned about chromatin in the previous video lectures, obviously. So chromatin modifications are the modifications or the addition of some chemical group on the chromatin. So before learning about chromatin modification, let's learn about what is chromatin. So chromatin is the nothing but the DNA which is wrapped up around the proteins. So those proteins are histone proteins, histones which are positively charged, over which the DNA, negatively charged DNA, is wrapped up, and that consists of the chromatin. So chromatin is the beads on the string structure. Okay, the beads on string structure which comprises of DNA as well as the proteins. Which proteins? The histone proteins, which are positively charged. Okay, the positively charged histone proteins, while DNA is the negatively charged. Why? Because the phosphate groups give the negative charge to the DNA. So chromatin has DNA as well as protein. So that is why chromatin modification consists of the modifications which are done on DNA, the DNA modifications, and other are histone modifications. Okay, we have already learned about DNA modifications. So we have learned about DNA methylation in brief detail. If you have not learned about it yet, go and watch the video lectures about DNA methylation, as well as I have also tried to discuss about the DNA demethylation. So in the DNA methylation, what happens is we add the CH3 group on the fifth carbon of the CpG dinucleotides. So on the CpG dinucleotides of the DNA, CH3 group is added on the cytosine of the cytosine on the fifth carbon of the cytosine on CpG dinucleotides. Okay, let me write it again. So we add CH3 group on the fifth carbon of cytosine of the CpG dinucleotides. Not any cytosine, only cytosine after which the guanine comes. Those are known as CpG dinucleotide. On this cytosine, on the fifth carbon, the CH3 group is added, and that is what we call as DNA methylation. So this DNA methylation is carried out by the enzymes known as DNMTs, DNA methyltransferases. We learnt about DNMT1, DNMT3A, DNMT3B, as well as the DNMT2, which is not actually DNA methyltransferase but RNA methyltransferase. It is also renamed as TRDMT1, which transfers the methyl group on the tRNA. So we have learned all of this till now, and DNA methylation leads to the repression. Okay, it leads to the silencing of gene. So whatever we are going to add on the chromatin, that addition of the addition of the some chemical moiety, some chemical group is added on the chromatin, and that added modification is known as mark. Okay, those are known as epigenetic marks. So the DNA, in the DNA methylation, it leads to the repression, means the silencing of gene, and that is why DNA methylation is also known as repressive mark. Why? Because it is leading to the silencing of gene. Okay, so that is a repressive mark. So this is about the DNA modifications, and we will learn about the histone modifications as well. So in chromatin modification, we have DNA modification as well as histone modifications. So the histone modifications are are of different type. The types which we are going to learn in details will be histone acetylation, Okay, so histone acetylation means addition of acetyl group on the histones. So on which histone, on which residue we will add, that we will also look about it briefly. So histone acetylation is always an active mark. Why? Because it, how does it do that? We will learn about it in detail, but for now, you just have to remember those things. That DNA methylation is repressive mark, while the histone acetylation is active mark because it leads to the activation of the genes on that chromatin. So this is all about histone acetylation. Next, we will be learning about histone methylation. Okay, yeah, I forgot to tell one thing. Histone acetylation is always done on the lysine residue. So, you remember when we were learning about nucleosomes, when we learned about the histones, how they are wrapped up, how they are, assembled in an octamer around which the DNA wraps, and that unit is known as nucleosome, which is the structural unit of chromatin. So, when we were learning about that, when we learned about histones, so the histones, uh, they have they have histone fold domain, okay, which is highly conservative across the it has been highly conservative across the evolution, from overall evolution. This histone fold domain is very crucial because it is going to provide the docking site for the DNA, and it has the C terminal extensions as well as N terminal tails. Okay, so those N-terminal tails, the long stretch of long stretch of amino acids which are protruding out of the octamer, these are known as N-terminal tails, and those are also known as histone tails. So all of the histone modifications are done on the amino acid groups, on the acid residues which are consisting in the N-terminal tails. So, let's assume that this is the histone, okay, and so imagine that this is an octamer, okay, so this is an octamer. The eight histones proteins are tightly, they are assembled together, and it is forming octamer. So we have learned about nucleosomal assembly as well. So this is the histone octamer over which the DNA is going to wrap around. So on this histone, okay, on this histone, it some N-terminal tails protrude out, which are known as histone tails. So on this histone, so these histone tails are, they are nothing but the stretch of amino acid. So we have different amino acids over this polypeptide stretch, and on those amino acid residues, the all of the chemical groups are added, and that chemical group addition is known as histone modification. Okay, so the histone acetylation, this modification, the acetylation modification is always done on lysine residue. Means addition of acetyl group is always done on lysine. So lysine is also written as K. So that is why we write histone acetylation as KAC. So this mark is written like this. So I will also explain how the marks are written. So histone methylation, just the way histone acetylation is done on lysine, histone methylation can be done on the two amino acid residues, lysine, okay, as well as arginine. Okay, so this can be done on lysine residues as well as arginine residues. And again, you can add only one acetyl group in the histone acetylation, but histone methylation can be monomethylation, dimethylation, or trimethylation. Means you can either add one methyl group, you can add two methyl groups, or you can add three methyl groups. Okay, and it is written in this way. Or you can add it on the arginine as well. So this is how we write. The arginine methylation is written as RME, while the methylation which is done on the lysine group is written as KME. On which histone it is done, we will take a look at it in the in a chart. Then the third histone modification is histone phosphorylation. Okay. So the way histone acetylation was active mark, histone methylation can be active mark, or it can be repressive mark. depending on which residue the methyl group is added. Again, how many methyls are added, whether it is one methyl or two methyl or trimethylation, depending on that, again on which histone it is going to be added, H3 or H4. It depends whether on that location of addition, it decides whether it will be active mark or repressive mark. We will learn about all those different marks, active marks as well as repressive mark, when we will discuss about histone methylation in detail. So histone acetylation is always an active mark, so let's mark it in a different color. Histone acetylation is always active mark. Histone methylation can be active or repressive mark, while histone phosphorylation, okay, histone phosphorylation, this is done on the serine or threonine residues, and it is written as SPH or TPH. This is how we write it. Okay, so the next modification is histone ubiquitinylation. Okay, histone ubiquitinylation means addition of ubiquitin group. The way we add the methyl group, or the way we add the acetyl group on the histones, we can add a ubiquitin group, which is a polypeptide, which is, I guess, about 76 amino acid or 77. I am not sure about this. I'll write it in the comments. So what's the exact number? So this is the moiety which is added, and that is why it is known as histone ubiquitinylation. The next type of the histone modification is histone sumylation. Okay, the addition of sumu group on the histone. So these are the different types of the histone modification. Let's I found this nice chart in this paper which was published in 2007. If you want, I can put the DOI in the comment section below. So this is discussing the all of the histone modification in brief. So let's learn about them in this group. So as I told you, acetylation, histone acetylation is always done on the lysine group. So that lysine could be on the H3 or H4, or it can be on the H2A or H2B. And it is always leads to the activation of genes, and that is the acetylation is an active mark. So the lysine, the ninth lysine residue, or 14, 18, or 56, those can be of the H3 can be acetylated, and the fifth, eighth, 13, or 16th lysine residue on H4 can be acetylated. So this is very specific. You don't have to learn all of this. You will just have to learn those active or repressive marks. So in the case of acetylation, only active marks, or in the case of methylation, active as well as repressive mark. You have to learn about this, certain of them, like H3K27 methylation, the trimethylation mark, or H3K9 trimethylation mark. All of those, or H4, H3K4 methylation mark. You will have to learn about all of this when we'll discuss in detail. So just now, you can just remember on which residue the modification is occurring, and whether it is activating mark or repressive mark. Let's not worry about on which histone and on which residue it is going to be added, because we will learn about this in detail in the upcoming video lectures. So for now, just focus that acetylation is done on lysine residue, and it is an activation mark. So it is written as KAC because lysine is written as K. Phosphorylation is done on serine and threonine, and that is why it can be written as SPH or TPH, and it leads to activation. It always leads to the activation of the chromatin, and it is done on the it is done on serine threonine residues on H3 or H2A or H2B. Okay. So the methylation can be done on arginine as well as lysine. Okay, and it could be depending on the lysine, it could be either activation or repression mark. But whenever it is done on arginine, it is always an activation mark, and this is written as RME if done on arginine, and KME if done on lysine, because R represents arginine, K represents lysine. So the methylation of arginine can be done on H3 or H4, while methylation on lysine can be done on H3 and H4 on this following residues. Okay. So other is the ubiquitinylation. It is also done on only lysine residue. So you can see most of the modification marks are done on the lysine residue. There are a few which are done on arginine, serine, or threonine. Arginine methylation is done, serine and threonine the phosphorylation is done. The rest of them are on the lysine. So ubiquitinylation is done on lysine and it is written as KUB. K is lysine, UB for ubiquitin moiety, and it is done on the H2A or H2B on this residues. Okay, and it can lead to activation or repression. So on the 119th H 119th lysine of H2A, ubiquitin is added. So that is why it is written as H2A K K, sorry, it should be written as H2A K119 ubiquitination. Okay, this is written as it is. If the methylation is done, okay, so let me just erase this. Okay, if the methylation is done on the, if you have to write that methylation is done on the lysine residue of H3, the lysine which is on the 27th position. Okay, so how will you write? You will write H3, then you will write about the residue. The residue is 27th, or let's say it is the ninth residue. Let's say the ninth residue, and the methylation is done. So you will write H3K9 methylation, and if it is 3, it is trimethylation. Okay, let's write it like this. So you have to write that methylation is done on the ninth lysine residue of H3 protein. So you will write, and it is trimethylation. Okay, so you will write it like this: H3K9Me3 means trimethylation, and it is read as H3K9 trimethylation mark. How it is read? H3K9 trimethylation mark. You can write as H3K4 methylation mark. You can, so it is monomethylation. You can write H3K27 Me3. Okay, so it is H3K27 trimethylation. So this is how you write the marks. Okay, so this is the way you are gonna write it. Okay, so let's move on to the next mark. So ubiquitinylation is done on lysine. It can lead to either activation and repression depending on the depending on the position. The SUMOylation is also done on lysine. It is written as KSU. It is done on H2A, H2B, and it always leads to the repression of the gene. Okay, and there is one more which is not that much discussed. Okay, this histone modification we are not going to discuss in detail, but just know about it. So it is the isomerization of proline ring. So the proline is amino acid. Okay, so it is written as P isomerization. So ISOM, P isomerized proline, and it is always the proline on the third H3 on the 30 to 38th proline. So it depends on the which residue it is going to act. So the prolyl ring is isomerized and it can lead to either activation or repression. We are not going to discuss about this modification in detail, but we are going to discuss about this. So let's do a quick recap. Acetylation will always lead to activation. So there are a few things that you have to learn by heart. Okay, so acetylation will always lead to activation. Okay, and it is always done on lysine. The methylation can lead to is always activation if it is done on arginine, but the methylation which is done on lysine, it could be active mark or it could be repressive mark. Okay, then phosphorylation which is done on serine or threonine residue, it will always be the active mark. Okay, and the ubiquitinylation, it can be active mark or repressive mark, and it is done on it is also done on lysine and of the H2A or H2B. So it depends. It can be active mark or it can be repressive mark. The SUMOylation is always a repressive mark and it is done on lysine. So remember that all of the histone modifications are done on the histone tails. Okay, so those residues are the histone tail residues. All of them are done on histone tails, and all of them are done on lysine. but a few like methylation, which can be done on arginine, and phosphorylation, which can be done on serine threonine. So these are the two examples that you have to learn, which are different, and but the rest of them are done on lysine. And acetylation is an always active mark, so you have to learn which are the active marks and which are the repressive marks. So this is all about it. But here is a quick nice fun thing that I told you. All of the histone modifications are done on the N-terminal tails or histone tails, but there are a few which are done on the core residues, on the residues which are into the core of the histone, and that is the 79 H379 methylation. This is the only mark, H3K79 methylation mark, which is the histone core modification. This is not the histone tail modification, this is the histone core modification. So this is a very important one, so mark it. And that's it for today. From next video lecture, we will learn about all of those post-translational modification in detail, step by step. In the next video lecture, we'll learn about histone modification. Till then, see you. Bye bye.